Okay, hello everyone. Thank you for attending my seminar, my EAI seminar today. My name is Ben Pearce, and I am a postdoctoral fellow at Johns Hopkins University, and I do origins of life research. Specifically, I study how planets obtain biomolecules for the emergence of life. And I'm going to be sharing with you our theoretical work, the majority of which I did during my PhD at McMaster University in Canada, on the linking the production of hydrogen cyanide in early Earth's atmosphere through tropospheric rainout to biomolecule production in warm little ponds. And we focus for biomolecules on what is on the information molecule that's thought to make up first life, which is RNA. I'll just mention a few of my collaborators here below. Ralph Pudritz was my PhD supervisor, and Karan Malaverdikani and Thomas Henning are two of our key collaborators at the Max Planck Institute for Astronomy, who we run all our atmospheric models with. All right. So let's begin by placing the origin of life in a time interval. We don't know the exact date that life emerged on Earth, but we can constrain the date by two different boundaries. The early boundary we call the habitability boundary, and this is the earliest point when early Earth could have been habitable and life could have existed on the surface. And the later boundary is the biosignature boundary, and this is the earliest point in which there's compelling evidence that life already existed on the surface. So we did a review paper for Astrobiology back in 2018 where we constrained these two boundaries with all the available data that was present. So for example, the habitability boundary is constrained by the formation of the sun and the Earth, the moon forming impact, which would have reset the clock. If life emerged before then, it wouldn't have survived the formation of a magma ocean. And then the cooling of that magma ocean, formation of a crust and hydrosphere. And then there's a little bit of complication because we don't really know how bombardment would have affected life. So the habitability boundary is kind of poorly constrained to 4.5 to 3.9 billion years ago, quite the wide interval of potential habitability. But the biosignature boundary is a lot better constrained because in Greenland, in what's called the Isua supercrustal belt, which is aged at 3.7 billion years old, there are the oldest stromatolite fossils. And these are these layered structures you see in rock, which are formed by microbial mats. Essentially, you have these photosynthetic organisms that get covered by naturally occurring silts and sediments. And then once they get covered, they can no longer photosynthesize. They need the sunlight. So they migrate upwards and leave behind layer upon layer of this carbonate rock right here. So you get these cool domical structures, and there's no abiotic way that is known that can make these by just geologic processes. They also measured the delta carbon 13 signature, which is a proxy for life, because life prefers light carbon. It prefers carbon 12 over the isotope carbon 13 because it allows enzymes to work faster. So naturally, life ends up having a lot more carbon 12 than carbon 13, and these fossils are very light in carbon 13. So this is also suggestive that this rock formation was formed by life. So when we talk about the emergence of life, and in this talk when I discuss our atmospheric models, we're modeling this time interval in between 4.5 and 3.9 billion years ago during what's called the Hadean eon. So I want you to imagine a warm little pond on early Earth. This is a picture that I took at Lassen Volcanic National Park in California, just on a vacation. But I was finding it difficult to find a picture of a warm little pond online that had no vegetation around. So this one actually looks like it could be on the Hadean Earth. And these ponds are actually upwards of 80 degrees Celsius. So they are pretty good analogs for what we're thinking about during the Hadean eon. So the question you could ask, if this is the site where life emerged, is how did biomolecules get to this pond? And just as an aside, the reason why we study warm little ponds as sites of emergence of life is because they dry down. So they're not permanently wet like in the ocean, but they dry down, and this procedure of wetting and drying actually allows RNA to polymerize. You need to remove water to form the phosphodiester bond between two RNA monomers. So this is kind of a critical point and why these environments are so favorable for the emergence of RNA. So back to the question, how would this pond get biomolecules? That's the natural question that we ask in our research. Well, there's a few options. There's meteorites. Here's a picture of the Murchison meteorite, which is known to contain a vast inventory of biomolecules, including amino acids, the building blocks of proteins, nucleobases and ribose, the building blocks of RNA, and even fatty acids, the building blocks of cell walls. So, you know, you almost have everything you need to make a protocell from meteorites. There's also interplanetary dust particles. These are about 100 micron sized grains. They're essentially like small meteorites, but they would have gotten their biomolecules from ultraviolet radiation processes on the icy grain surfaces, whereas meteorites are likely to have gotten a vast majority of their biomolecules from aqueous processes inside asteroid interiors. So different kind of origins of biomolecules, but this is very much just another rock in space, just a very tiny one. The difference between IDPs and meteorites in terms of being deposited into ponds, though, is that IDPs are so small they just rain down constantly, and they bring their small picogram abundances of biomolecules very slowly, whereas meteorites are kind of this large milligram abundance of biomolecules that they deposit all in one event. So those are the two external sources, but if early Earth could have formed its own biomolecules, then whatever is rained into the pond forms up the pond solution, and this is what is going to allow aqueous chemistry to ensue and form biomolecules. So stuff like hydrogen cyanide is water soluble, will rain into this pond, and then you have radiation hitting the aqueous surface, and you have wetting and drying. So you have a few physical processes here which can help chemistry ensue, and that's really the question: is what kind of biomolecules could be produced in the pond versus were delivered to the pond. So this has been the main question in my research and our research for a long time: is where did these building blocks originate? How did they get to ponds? Now, this was first studied by Carl Sagan and Christopher Chiba in 1992, where they just compiled estimates of various different sources of biomolecules to the Earth's surface. And they found that interplanetary dust particles and atmospheric production dominated the amount of organic carbon which would have reached the Earth's surface. But what they didn't consider was where this organic carbon was being delivered. And it matters, because let's say you deliver the organic carbon to the ocean. Well, this is a huge voluminous body of water. Any organics you deliver there are simply going to dilute, and it's not going to be very useful for making biomolecules, for making RNA or life. So you need to care about where you deposit your organics. And this is why we study warm little ponds, because they can dry down, they have an opportunity to concentrate the solution and allow RNA to form through polymerization. So in my master's research back in 2015 to 2017, we computed, using a numerical model, the fate of nucleobases, which are these C, G, A, and U on this RNA molecule right here, from meteorites and IDPs to warm little ponds. Here's a schematic of that model. You can see right here how the model works. It's essentially a sources and sinks models, which is controlled by differential equations that have values for the sink and the source rates. And the rate data comes from experiments, historical precipitation record, or the lunar cratering record in terms of the mass delivered. So I'll just take you quickly through the different sources and sinks. For pond water, how does the pond wet and dry? Well, you got precipitation coming in, which varies sinusoidally over a season. And then you have evaporation and seepage, which is seeping through pores in the base of the pond. as your two sinks of water. So this allows for natural wet-dry cycles, as I'll show you in the next slide. For nucleobases, the sources we study are meteorites and IDPs, and then there are various sinks in the pond like hydrolysis, which is reactions with water. You got seepage. These things are also carried through pores in the base of the pond, and you have UV photodissociation, which blasts apart these nucleobases as well. Looking at just the pond water level on the left here, we have percent full on the y-axis and time on the x-axis. We studied a few different analog environments on Earth today, and we found there's a lot of degeneracies in our pond water models. For instance, Colombia at 65 degrees matched Thailand at 20 degrees. So instead of just trying to model all these different latitude and longitudes on Earth, we just chose 3 different environments which characterize 3 potential different pond conditions on early Earth. So we call them the wet environment, which never dries down completely, the intermediate environment, which has a 6-month wet and 6-month dry period, and the dry environment, which has a 3-month wet and 9-month dry period. When we look at the intermediate environment in this right plot here, this is the adenine concentration in the pond from interplanetary dust particles. And we can see when the pond, this is the pond water level again in blue. When the pond dries up, your adenine reaches its highest concentration, and then UV dissociation blasts away until adenine reaches this flat top here. And this is when the incoming adenine rate matches the outgoing adenine rate. Then the pond fills again and the adenine dilutes. And this pattern continues, and it never increases in concentration. And this is PPQ, that stands for parts per quadrillion. This is negligible. So what this tells us is that you're never going to be able to concentrate your nucleobases from interplanetary dust particles because they rain down on the surface too slowly, that these sinks, these destruction processes are too efficient at removing them. So you just cannot accumulate any nucleobases from IDPs. But what about meteorites? In this plot here, we in red and blue for the intermediate and wet environment, we compare meteoritic concentration of adenine to IDP concentrations. And this is a vast difference. Look at this. That's like 10 orders of magnitude at peak. So the huge difference here is that meteorites, they enter the pond. Diffusion is relatively quickly. We model diffusion of nucleobases out of the pores of the meteorite. It only takes about a week. So you reach parts per million concentrations, but it's a finite source. Unlike IDPs, which are raining down constantly, meteorite depositions are single events. So you reach parts per million for a few weeks, and then you begin to, the sinks in the pond begin to remove your adenine. This is when UV was turned on. Here you see this cliff, and in the wet environment, UV is never turned on, and seepage is the next most efficient sink. So seepage slowly carries away the adenine out of the pond. So this was interesting to us because here we have a plausible source of the building blocks of RNA from meteorites, but it's finite. And this really made us a little uncomfortable because it doesn't give you a lot of time for life to form, for RNA even to form, but then eventually for life to form through the process of evolution of these RNA species. So we thought, well, how does this compare to in situ production? What could Earth have produced on its own without this external delivery? And this is what we decided to study in my PhD. And in other words, is meteoric delivery necessary for the origin of life? So this story now takes a turn into hydrogen cyanide production in the atmosphere, as this is key for biomolecule production in ponds. Here, the experimental grounding of our theoretical research is the Miller-Urey experiment. Here is Stanley Miller looking at his original apparatus that he used when he was a grad student. And what he did originally was he put a bunch of reducing gases like hydrogen, methane, ammonia, and water into this apparatus and exposed it to a spark discharge to simulate a lightning strike. Hydrogen cyanide was then produced in that flask here, and then it's condensed out and rained down into this reservoir here, into this pond, if you will. They then measured the solution here for biomolecules, and they found about 6 amino acids in the original experiment, which was very exciting because this was in the 1950s, this was the first time that biomolecules were made in a primeval atmospheric setting from just purely abiotic physical processes. So this really kicked off the origins of life field in a big way. This experiment has since been also done for nucleobases, for the U, A, G, and C in RNA. So what they found that was kind of interesting in this experiment is they redid it with a oxidizing atmosphere, mostly CO2, and it didn't work as well. They got about 4 orders of magnitude lower yields of amino acids from the oxidizing atmosphere. And this is a little foreshadowing because it's going to come up in our theoretical models. You know, why is it that a reducing atmosphere is favorable for hydrogen cyanide production and for biomolecule production, but oxidizing conditions aren't as favorable? So just a few things that hydrogen cyanide could make in the pond. They're a prebiotic source of nucleobases. They can be irradiated to make formaldehyde, which through the foremost reaction can make ribose. They're involved in amino acid synthesis and 2-amino-oxazole synthesis, which is another precursor to nucleotides. And what's interesting here about hydrogen cyanide is that it doesn't really matter. There's a lot of debate in the field on, how do you actually get the entire nucleotide, the C attached to the ribose and the phosphate. There's a lot of debate on how you do that. Some that don't even involve the nucleobase, but it doesn't matter in our research because hydrogen cyanide is a precursor to all of these prebiotic routes to nucleotide production. So HCN really is an important precursor for life. So we model atmospheric chemistry, and what do you need to model atmospheric chemistry? Well, you need chemical data, you need your chemical network, which is a collection of reaction rate coefficients as a function of temperature and pressure. And you need an atmospheric code, which handles the rate of transfer from the star through the atmosphere, through the atmospheric layers, diffusion of species through the atmospheric layers. You got photodestruction, galactic cosmic ray chemistry, and it also runs your chemical network. This is where our collaboration with the Max Planck Institute for Astronomy comes in. We use ChemCam, which was developed by Karan Malavertakani and Thomas Henning and Paul Moliere. And for our chemical data, we use a combination of experimental rate coefficients when they're available, but in the vast majority of cases, we calculate them using quantum chemistry, using a consistent quantum chemical method. And as it turns out, there's a lot of unknown chemistry, even with hydrogen cyanide and formaldehyde, that we were able to rectify. We call our network Kraken because it is a consistent reduced atmospheric hybrid chemical network, and it's got HCN there in the title because it is a HCN focused network. So how do you use quantum chemistry to calculate reaction rate coefficients? I'm going to teach you in one slide. So here, just to show you, we began with a very reduced network that only involved species from an atmosphere composed of hydrogen, methane, and nitrogen. And when you destroy those species, when you break them apart, you get some of the fragments you see here in this little GIF here, such as CH3, ground state and excited state CH2, atomic nitrogen in the ground state and the excited state. And these all react differently when you do the quantum chemical simulations. Anytime you see a line between two species, that's when I was able to get those species to react on a potential energy surface. And here is an example of one of those reactions. Here you have methane reacting with hydrogen. What I did was I froze the distance between H1 and H5, these two hydrogen atoms, and then I allowed the geometry to relax down to its lowest energy state. And when I plot the distance between the hydrogens, or the reaction coordinate, versus the energy, I get a plot that looks like this, which has a big bump in the middle. And at the top of that bump, we call it the transition state. This is the highest energy state that the species must overcome to get from reactants, which is methane plus hydrogen, into products, which is CH3 plus hydrogen, the hydrogen molecule. Then I use the principles of statistical mechanics to calculate the reaction rate coefficient. So we use what's called canonical variational transition state theory. If you have taken a StatMech course, canonical is probably a word that you recognize. There's also micro-canonical and grand canonical. So I take the partition functions and the energy of the transition state and the reactants, and I can calculate how frequently that reaction will occur. As you can imagine, the larger the barrier, the less often that reaction is going to be able to occur in nature. And for the quantum chemists in the room, we use a density functional theory functional called BH&HLIP, which we found to be the most consistently, the most accurate when comparing to experimental data. What was very exciting was that in this initial study, we discovered 48 previously unknown reactions that have not been reported in the literature. And I was a PhD student at the time, so that was really exciting for me because on a daily basis, I was discovering new reactions. And I had this big whiteboard and I just write down a new reaction every time I discovered it. I didn't know how important they were, but I knew they existed. And I could calculate their rate coefficients. When dealing with new chemistry, you must validate it. So this is where our study of Titan's atmosphere comes in. The species involved in that were just hydrogen, methane, and nitrogen and their dissociation fragments. And this is why Titan is a good place to do validation, because it's a very simple atmosphere of nitrogen, methane, and hydrogen with very few oxygen species. The dominant one is carbon monoxide, which is pretty inert. So by not including oxygen in this initial validation, it didn't really lead to very much error. Titan is also the most similar atmosphere to early Earth in our solar system, as it is very reducing. It's very cold, not the same temperature, but definitely similar in pressure and composition to the earliest stages of the Hadean atmosphere. One thing unique to Titan, being a moon around a gas giant, is that Saturn's magnetic field lines actually precipitate galactic cosmic rays down into Titan's upper atmosphere, which collide with the upper atmospheric species and then send electron showers down to the lowest part of the atmosphere. And those electrons collide with molecules and dissociate them, similar to how photochemistry works. Instead of a molecule absorbing a photon and dissociating, exciting and then dissociating, an electron could collide with the molecule and excite it to dissociation. And the reason why we can use Titan to validate is that Cassini, the spacecraft, went to Titan in the early 2000s and measured hydrogen cyanide at various altitudes with 4 different instruments. Here are our models of Titan's atmosphere. You can see altitude of Titan's atmosphere on the y-axis and the hydrogen cyanide abundance on the x-axis. The Cassini data points are in yellow, blue, green, and purple. You can see our fiducial model in black goes right through these yellow data points in the lower atmosphere, then goes through the first few blue and green data points here, and then veers to the left of these green and blue data points, but goes right through this purple data point, which happens to be the only in situ measurement of hydrogen cyanide in Titan's atmosphere. This is when Cassini dipped down to 1050 kilometers in Titan's atmosphere, took a gas sample and put it through a mass spec to measure its composition. So we were pretty stoked to have our model just happen to go through that data point, as it's likely the most accurate. And this just shows you that our chemistry is doing a pretty good job at representing hydrogen cyanide production in Titan's atmosphere. You can see on the right here, the 3 most recent Titan models in the literature in orange, blue, and pink. And this just shows you we're right in line with the state of the art. Now, one other thing, this red dotted line right here is called Kraken 19. We did a sensitivity analysis where we removed 1 reaction at a time to try and reduce our atmosphere, our network, which had over 100 reactions, even further. And if the removal of the reaction didn't cause the hydrogen cyanide profile to change, we just removed it from the network and continued the removing reactions one by one. And when we did that, we found it all boiled down to 19 reactions that were responsible for hydrogen cyanide production and destruction. So if you're on the left here, this is a sensitivity analysis where the central line is the hydrogen cyanide profile from our fiducial model. If the removal of reaction causes your curve to be on the left of this, that means that less hydrogen cyanide was produced when you were removed. In other words, you were involved in hydrogen cyanide production. And if you're on the right, then you're involved in hydrogen cyanide destruction. I'll just highlight this yellow one that's going off the page because it's clearly very important. This is cyanide plus methane, goes to hydrogen cyanide plus CH3. This is clearly the dominant mechanism of hydrogen cyanide production in Titan's atmosphere. Well, Titan's a lower atmosphere, I should say. All right, so this reducing the network to 19 reactions allows us to analyze those reactions, tell a story of how hydrogen cyanide is produced, and I'll tell you that story now. It begins with ultraviolet and galactic cosmic ray dissociation of the primal species, the primary species in the atmosphere to produce reactive radicals. Those reactive radicals are processed, and then hydrogen cyanide is produced along 3 main channels here, which are nitrogen atoms either in the excited state or the ground state, reacting with methane or CH3. Then hydrogen cyanide, once it's produced, photo dissociation occurs and pops off that hydrogen. But as you saw on the previous slide with the yellow reaction going off the page, cyanide plus methane is the dominant process to recycle that cyanide back into hydrogen cyanide. And that ends up accounting for 36 to 46% of the total hydrogen cyanide produced in Titan's atmosphere. You of course need cyanide in the first place for this channel D. So in order to get HCN originally, it's really channels A, B, and C that form the initial hydrogen cyanide in the upper atmosphere. And what's really exciting is that channel C was previously undiscovered until we did our initial exploratory quantum chemistry study in 2019. So this somewhat validates us going through the quantum chemistry, discovering these unknown reactions and adding them to create a more accurate network for hydrogen cyanide chemistry. Okay, let's get to the juice of this talk, which is modeling early Earth's atmosphere. Again, we're using ChemKM, our collaboration with the Max Planck Institute for Astronomy, but we had to upgrade our network because we know early Earth had oxygen species such as CO2 and water and the fragments from dissociation of those oxygen atoms and the hydroxyl radical. And also we want to study formaldehyde as a potential atmospheric species. So we expanded that network and we ended up doing another exploratory quantum chemistry study, and we discovered another 48 previously unknown reactions that produce hydrogen cyanide and formaldehyde from oxygen pathways. We do not have a pressure temperature profile for early Earth, so we have to calculate it, and we use a radiative transfer code called petit radtrans. And this allows us to be consistent with the composition and the pressure temperature profile, considering the greenhouse gases that would have been present and the various opacity sources. We modeled 2 different epochs during the Hadean eon in the time interval for the origin of life, one at 4.4 billion years ago and one at 4 billion years ago, which represent hydrogen and CO2 dominant phases, or reducing and oxidizing phases. And these epochs vary in the bombardment rate, which we can tell from the lunar cratering record. They vary in luminosity. We know the sun increases in luminosity over time. UV intensity and lightning chemistry are also different. And then we couple hydrogen cyanide rainout from those models with the warm little pond model that I showed you in the beginning part of this talk, where we modeled meteorite and IDP concentrations in ponds. Here is a graphic summary of our atmospheric model, focusing on 4 key species. Hydrogen and CO2 control whether your atmosphere is reducing or oxidizing. Methane is the main precursor to hydrogen cyanide, and hydrogen cyanide is really the star of this show. So we have it listed here as the sources and the sinks for each of these species in our atmospheres. And if it's a source, that means that we're inputting that species into, in all cases here, it is the lowest layer of the atmosphere, unless it's chemistry, and then that's occurring from the chemical network in all layers of the atmosphere. For hydrogen, the main source is impact degassing. So we have the bombardment rate. We know how much material was coming in, and we know that it was likely primarily in statite meteorites, which contain a lot of iron. So that iron would have reacted with water on the surface and in the atmosphere to produce iron oxides, and it would have released hydrogen. And this is how you get reducing atmospheres. You have a lot of bombardment, and you can oxidize a lot of iron and release a lot of hydrogen into the atmosphere. But that hydrogen is really light. And if your atmosphere is very hot, it escapes to space quite efficiently. So there's a balance between impact degassing and escape to space, which create your reducing or oxidizing conditions, depending on what your rates are that you're using. For CO2, the main source is volcanism. We know from zircon analyses that the mantle was oxidized as early as 4.4 billion years ago. So it would have been CO2 coming out of volcanoes, not H2. And then CO2 is water soluble. We know the carbon cycle, so it rains out of the lowest layer of our atmospheres. For methane, we consider the methane source from hydrothermal systems. This is when ultramafic rocks react with water in the ocean floor to produce hydrogen gas. And then that hydrogen reacts in the aqueous environment here with CO2 coming from the vents through a process called Fischer-Tropsch synthesis to produce methane. And then that methane can be released into the atmosphere. The main source for hydrogen cyanide, as we saw also with our Titan study, is really the dissociation of species such as nitrogen and methane, either from UV or lightning in the case of early Earth, to produce radicals which react to form hydrogen cyanide. That hydrogen cyanide then rains out of the lowest layer of our atmosphere, and that's how we connect it to our pond models. Okay, here are the calculated pressure temperature profiles. And you can see we modeled 2 different reducing models and 2 different oxidizing models with 2 different surface temperatures. So we used a hydrogen dominant for 4.4 billion years ago, and changing the methane from 1 to 2% increased the surface temperature from 50 to 78 degrees Celsius. So you can see we're already bordering on uninhabitable here. You know, we get much higher, we get up to 100, and you're not going to have any ponds. You're not going to be able to condense out your water to form ponds, to have wet dry cycles, to form RNA. So there is a limit here to how much methane we can allow into the atmosphere in this H2 rich environment. And the main reason is what's called H2H2 collision induced absorption. This is when 2 hydrogen species, 2 hydrogen molecules come, they're in a high pressure environment, they come close enough together that they form this essentially supramolecule, which has a different absorption bands than just the species on their own. And they absorb in the infrared when they're that close together, which creates a greenhouse effect. So we found that when your atmospheric pressure gets higher than about 1.13 bar, H2H2 collision induced absorption starts to become very efficient and takes your atmospheres out of habitability if you have too much methane, which is also a very efficient greenhouse gas. But if you're in an oxidizing environment, you don't have that H2H2 collision-induced absorption. CO2 is not as efficient of a greenhouse as H2 in that high pressure environment, so you can have a little bit more methane. We went up to 10% here to get a 50 degrees Celsius surface for this 4.0 billion year ago model. Okay, here are our atmospheric models. We have altitude of early Earth's atmosphere on the y-axis and the abundance of 2 species on the x-axis. We did both formaldehyde in solid lines and hydrogen cyanide in dotted lines. The reason we did formaldehyde is because we weren't sure at the time whether formaldehyde, which is also a key species for biomolecule production, we weren't sure if it came from the atmosphere or if it could have been produced in the pond from hydrogen cyanide. And what we found out here is that it's not really coming from the atmosphere during the reducing phase. These are 10 to the minus 16, 10 to the minus 15. This is really not enough to rain down into the pond. Hydrogen cyanide, on the other hand, gets up to 10 to the minus 10 here after 15 million years. So this is sufficient to rain down into the pond and accumulate. These 2 reducing models here produce much higher HCN abundances than the oxidizing models on the right. We can see on the right, it's about 10 to the minus 15 at the most. So there's about 4 or 5 orders of magnitude less hydrogen cyanide produced in the oxidizing phase than in the reducing phase. And this is what's exciting because going back to the Miller-Urey experiment, they found that biomolecule production during in a reduced atmosphere is about 4 orders of magnitude more efficient at producing amino acids. So this could be because hydrogen cyanide is produced less efficiently in the oxidizing environment. If we look at just the surface abundance of various species. So just that bottom atmospheric layer. And I'll just point out methane in pink and hydrogen cyanide in purple. We see there's a correlation. We see a few different correlations here. So for instance, looking at the pink and purple, after about 100 or 300 years or so, these lines kind of trace each other in all of these plots. So right after this point. So there seems to be a correlation between, having a stable amount of methane, this is 10 to the minus 6, and having a lot of hydrogen cyanide. So we see here when hydrogen cyanide goes down, dips down here due to the hydrogen abundance decreasing, we have a lot less hydrogen cyanide here at 10 to the minus 15. We can also see an anti-correlation. This blue line right here is oxygen. It kind of has the same opposite shape as the hydrogen cyanide. So in a highly oxidizing environment, hydrogen cyanide, which is a reduced form of carbon, is going to be produced in less abundances, and formaldehyde, which is a oxidized form of carbon, H2CO, is going to be produced in higher abundances. So there's a kind of an anti-correlation between hydrogen cyanide and oxygen, and a correlation between oxygen and formaldehyde. So what we see here is that kind of the takeaway message from here is that you need a stable source of methane, and you need your methane to stick around. And it really only sticks around in high H2 environments. If you have low H2, your methane doesn't stick around. That carbon gets oxidized, and then you don't produce as much HCN. And this is why you produce more hydrogen cyanide in reducing environments. We wanted to know whether it was lightning or UV photochemistry, which was producing most of our HCNs. We turned each one of these off one by one. On the left plot here, we turn off photochemistry, and on the right plot, we turn off lightning. And we see that if you don't have photochemistry, you get about 9 orders of magnitude less hydrogen cyanide than you do if you turn off lightning. So it's really not lightning that's producing HCN in our models, it's photochemistry. And it's occurring mostly right here at about 500 kilometers up in the atmosphere. And it's being mixed down to the lowest atmospheric layers here from a process called eddy diffusion or turbulent mixing. Okay, here we come to the main plot of this talk. This is the big comparison. We're going back, this is our pond model. We're gonna go back to meteorites we see here in brown, IDPs we see now in purple, and this was the original plot I showed you in our early models. But now we've added in situ production of adenine in our ponds from the reducing phase at 4.4 billion years ago and the oxidizing phase at 4.0 billion years ago. And what we see when we compare these is that the reducing phase is similar in composition to what meteorites would have delivered after about a few weeks. There's about a 3 order of magnitude difference between the peak abundance of adenine from meteorites and the peak abundance of adenine from the reducing phase from hydrogen cyanide. And the key difference here is that the in situ production lasts for over 100 million years instead of just a few weeks. So although these concentrations are 3 orders of magnitude lower, this is your base abundance. This is what you're always going to be able to have in your pond during that 4.4 billion year ago phase. Whereas meteorites are going to only provide bursts in nucleobase concentration above the base phase. We can see this huge difference between the oxidizing and reduced concentrations of adenine, about 4 orders of magnitude. This just really shows you by the time the atmosphere oxidizes, in situ production really isn't a plausible source of biomolecules. It's really only during this reducing phase and from meteorites. We have a bunch of other biomolecules here on the right: guanine, cytosine, uracil, thymine, the other nucleobases, 2-aminoxazole, ribose. And what's interesting here is that a few of these are not present in meteorites. So this is a plausible source of a few of the other building blocks of RNA that you can't get from meteoritic delivery. So your cytosine, your thymine, for instance, and your 2-aminoxazole. The formaldehyde, I know I mentioned to you that we were interested in see whether formaldehyde could have come from the atmosphere. Well, formaldehyde from rainout is this dotted line here, and formaldehyde from aqueous production, which is a process where UV light can irradiate hydrogen cyanide in solution and produce formaldehyde. That produces about 3 orders of magnitude more formaldehyde than rain out. So formaldehyde likely came during the reducing phase from UV pond chemistry rather than from direct rain out from the atmosphere, which we didn't know before we did this study. Okay, so let's look at these concentrations. Let's just talk about this a little bit more. This is nanomolar, or parts per billion, and this is pretty low. If you think about laboratory experiments which produce nucleotides from nucleobases or from hydrogen cyanide, they're really using hundreds of micromolar, millimolar, or even just molar concentrations. So they're much higher concentrations used in the lab. And there's a reason to do that. Of course, in the lab, you need higher concentrations because you need to be able to measure small yields. But is there a way for us to concentrate these ponds higher so that we get closer to those laboratory concentrations? Well, one idea we had, and this is the original hypothesis by David Deamer and Bruce Damer, is what if your pond was clogged? What if the pores at the base of the pond were clogged up by something like amphiphiles? So for instance, meteorites deliver fatty acids to ponds. These amphiphiles can connect together to form matrices, and they could block any biomolecules in the dry phase from seeping through those pores in the base of the pond. So we turned off seepage in our models to see what kind of concentration enhancements we can get from this. And we see about 1 to 3 orders of magnitude increased concentrations from this no seepage model. The next most dominant sink is hydrolysis. So that ends up being the concentration limiting sink after seepage is turned off. So we get up to about 10 parts per billion of adenine. Guanine and cytosine reach steady state very relatively quickly, but uracil and thymine have very low hydrolysis rates, so they actually can increase to higher concentrations up to the molar or the micromolar concentrations here after about 10,000 years. So we found this interesting. Here is a summary of that plot. We have our 5 nucleobases here. We have the concentration in the pond from the peak concentration from the early Hadean in brown, the late Hadean in blue, and then this no seepage model in yellow. And we can see we get up to just about micromolar with the no seepage model. These are still quite low concentrations, but there could be possible ways to concentrate this further. For instance, if you're encapsulated into a vesicle or a mineral gel or something like this. Okay, so this is essentially the limits to biomolecule production in ponds from atmospheric chemistry. So let's I'll just go over a few takeaways here. We find that it's really atmospheric methane which is the driver of hydrogen cyanide production. But we find that if you have too much methane in a hydrogen-rich atmosphere, you become uninhabitable. So you can't just increase methane production on early Earth and have more hydrogen cyanide production in the atmosphere for biomolecule production in ponds. There's a limit because you need to maintain habitability. You need to be under 100 degrees Celsius to have the pond to rain down into. We find that the limits of RNA building production for habitable Hadean surfaces is in the nanomolar to micromolar range, but meteorites can provide boosts up to tens of micromolar for a few weeks. And then finally, because the atmosphere switches from reducing to atmosphere to oxidizing conditions when the bombardment rate decreases, we find that this work points to an early origin of RNA within about 200 million years of the moon forming impact, which if we go back to our time interval for the origin of life, that would be right here from about 4.5 to 4.3 billion years ago. So this is a very early origin of life in this habitable boundary range. I'll just finish up with one slide here to tell you what I'm up to now. So that work I shared with you was mostly done during my PhD. And I'm now a postdoc at Johns Hopkins University, and I've switched to experiments. So I'm interested in taking the theory that I've worked on and adding some ground truth to it by doing atmospheric chemical experiments using the phaser chamber. And you can see here, picture me in front of the instrument. This is the main chamber. Essentially, we can put any atmospheric gases we want in this chamber and expose it to lightning and photochemistry, and see this gas flows into a mass spec right here, and we can read out how much hydrogen cyanide we're producing. So this is the experiments I'm working on now with Professor Sarah Hurst, who's the PI of the lab, and Dr. Chao He, who put together this instrument about five years ago now. So I'll be excited to share these experiments with you sometime later in 2022. And with that, I will say thank you for listening, and I'll take any questions you have.
I'll just start. So, hi Ben,
Hi.
Alex. Hi, Alex Roof from LMU Munich. So, very nice talk. Thank you very much. I would have a question. So HCN is like very important for like probably like kind of all prebiotic building blocks. Do you have like an educated guess for where phosphorus comes from, especially like when discussing atmospheric chemistry?
Yeah, that's a good question. It's not really something that we have focused on in our models yet, because there are a lot of plausible sources of phosphorus for early Earth for early Earth ponds. One main one that we sometimes talk about is ribosite, which is a reduced form of phosphorus that can come from iron-rich meteorites. It is true, like you can't just have any source of phosphorus to phosphorylate your nucleosides for nucleotide production. You know, you really want some sort of reduced form, such as ribosite. But who's someone? Matthew Pasek is someone who's really studied this in great detail, and it's definitely something that we'll need to eventually add to our models. But it wasn't the biggest hurdle.
So in the sake of time, I just may add on like one more. I was just curious if you could like think about, you know, like very provocatively asking like phosphine chemistry or any like atmospheric type of phosphorus chemistry, or like, do you think that's an option to add on or not? Maybe
That's
like more to the
interesting.
minerals.
Yeah, it's interesting. This is actually something that I wrote a proposal to do because phosphine chemistry is relatively underexplored in terms of the reaction rate coefficients for chemistry, for atmospheric chemistry. Actually, in that original Venus phosphine modeling paper, they didn't even have really very much phosphine chemistry. So they took analogous nitrogen chemistry. You know, they took like ammonia, and they said, well, let's just say that all the nitrogen reactions have a analogous phosphorus reaction, and we'll just use those rate coefficients. So it's not really as accurate as you would like. So I wrote a proposal to uncover all this unknown phosphine chemistry so that you could do these sorts of studies, but I haven't started that work. But that would be the first order of business, I would say, to try and answer that question, because as it stands, using the analogous nitrogen chemistry, it doesn't really seem like phosphine is made in very high concentrations. But we don't know. I mean, with all this unknown chemistry, whether that's fully true or not. So I would really love to do an exploratory work for that.
Right, yeah, I think it's interesting to have an alternative source, because like all the minerals, they have problems with leaching and all that stuff. So I'm curious to get news from it. So thank you again
yeah,
for the talk.
No worries. Thanks for your questions.
Hi, Ben. This is Augusto Carballido from Baylor University in Texas.
Hello.
Hi. Yeah, thank you for that talk. It was very informative. So my question is regarding, I want to see if I understood one of your takeaways correctly. So do we understand that the meteoritic delivery of organics only played a minor role in Hadean?
Yeah, I guess what we're seeing is that it can provide bursts in concentration, but because it's a finite source, so for instance, let's say you have an asteroid entering the Earth's atmosphere, or a fragment of an asteroid entering Earth's atmosphere. It's going to fragment along pre-existing cracks, and then it's going to start to spread out, and it will reach terminal velocity before it gets to the surface, and it'll spread out its fragments in what's called a strewn field. And so these are non-impactors. These are just fragments falling on the surface, like the ones that we pick up in Antarctica and across the planet today. So these things, the overlap of the strewn field with a pond can occur, but that is only going to occur probably once to one pond. For any given pond, it's only going to happen once from 4.5 to 3.7 billion years ago. So you really only have one deposition event per pond on early Earth. So it's really a finite source. There's no, you have to do everything you can with what you're given in that pond that one time. So with all the sinks in the pond, it depletes within weeks. So it's not necessarily that meteorites are not a key source, because they can provide boosts up to 3 orders of magnitude in the concentration. But because they're finite, it doesn't really seem like the entire story. What we see here with atmospheric production of hydrogen cyanide and rainout is the base background amount of biomolecules is a lot more constant and can bring certain biomolecules that are not present in meteorites to the pond as well, albeit in lower concentrations. So I think that maybe both are important. But if I had to choose one over the other, having the constant source from atmospheric production is a lot more plausible in my mind, as it would seem that RNA production needs a lot more than a few weeks time in order to produce a lot of different diverse strands of RNA to get the ribosome or the ribozyme, and then eventually something that we would consider to be living.
Yeah, and these meteoritic bursts would occur in the early or in the late Hadean.
Yeah, that's right.
Okay.
Yeah.
Thank you.
No problem.
Ask like one more question, as we
Absolutely,
are like still in time. It's maybe like a bit of a dumb one, but I didn't probably get it. Like on slide 28, like on your last paper. So do you see these 4 different compartments as being isolated or connected? So I just wondered if you include also like redox reactions like H2 plus CO2, or sorry, it's slide 28.
28, yeah. So I was just
I just wonder if like H2 and CO2 can like vanish themselves out, or are they, I don't know, so high in concentration that they are probably like quasi-stationary, or like. So do you expect H2 plus CO2 or methane plus CO2 or not?
We essentially there's going to be a little bit of both, but we see that one really tends to dominate. Actually, what we end up seeing, which is not even on here, is nitrogen ends up being the gas that just sticks around in our models and ends up after 100,000 years or 100 million years, sorry. Nitrogen is really the dominant species at usually about 70, well, usually about what percentage it's at today, which was pretty interesting, but it makes a lot of sense because there's not a lot of sinks for nitrogen. But what we started with in our models was either hydrogen dominant or CO2 dominant, and then we let the chemistry and the sources and the sinks play out. And in no cases was there both hydrogen and CO2 in high abundance with our source and sink rates. There probably would be some perfect impact degassing where you could get somewhat similar concentrations of both, but yeah, it's because we modeled such different phases, 4.4 and 4.0, which had very different bombardment rates. They ended up being hydrogen dominant or CO2 dominant as the secondary species to nitrogen.
Okay,
Methane, though, from hydrothermal systems never really got above the parts per million, the few parts per million concentrations in these models, which is not a problem, because if it was too high, then your surface would no longer be habitable. There are some other models out there with ideas that, if you just increase the methane, that you can get more hydrogen cyanide production, but they don't do their radiative transfer modeling to get their pressure temperature profiles. So it would be, there would be a limit to how much methane you can get to still be habitable, and it's not very high.
Okay, I see. Thanks.
No problem.
Maybe just like one question in terms of perspective. So do you think you can like apply your model to any kind of atmospheric systems, like speaking about exoplanets, like in future,
absolutely.
when more data come to that? Or
yeah, the postdoc Karan Mullavertikani, who was at Max Planck Institute for Astronomy, is now at LMU. For this research, developed a lot of different functionality for this code, including outgassing of species and rainout of species. So basically, like increasing the amount of sources and sinks. You can include lightning chemistry, all this kind of stuff. So it's a pretty diverse model now that you can apply to really any terrestrial planet. We've done galactic cosmic rays. There's almost nothing that I could think of that we could. I mean, there's always something you could think of to add, but especially with an exoplanetary system where, I guess tidal locking, that's something that's not really considered in this 1D model. So maybe that's something that could be added for M-dwarf exoplanetary systems. But for the most part, yeah, it could be. You could do. It'd be really interesting to do an exoplanetary study where you have more theoretical bombardment rates, which are not kind of linked to our lunar cratering record, but that could be taken from, say, protoplanetary disk modeling results and body simulations or something like that. I think there's a lot of opportunity for more exoplanetary works, for sure.
Great, yeah, check that out in future. Thank you. Thanks, Ben.
Cool.